In this module, we'll discuss the endocannabinoid system. The endocannabinoid system plays many critical roles in biological processes that are relevant to diverse areas of medicine, ranging from psychiatry to immunology. Lots of research has established the role of this system in maintaining general homeostasis or balance. In the brain, for example, endocannabinoids modulate neuronal conversations. They keep them from becoming too loud and out of control. The endocannabinoid system is comprised of three components. First, there are the endocannabinoids themselves, and the body makes these from lipid precursors, and there are two of them, anandamide and 2-arachidonyl glycerol. These are the most well-researched and abundant endocannabinoids, but it's very likely that there are others that have yet to be characterized. The second component consists of their receptors, which include but are not limited to CB1 and CB2. Both of these receptors are expressed throughout the body, but CB1 predominates in the central nervous system, while CB2 tends to be abundant in the peripheral organs. The third component consists of enzymes that tightly control the synthesis and degradation of endocannabinoids. This is a schematic showing how endocannabinoids work in the central nervous system at the level of a synapse. The CB1 receptor predominates here in the CNS, and it functions to restrain excessive neurotransmission. So a neurotransmitter binds to its receptor, and that binding triggers the synthesis of the endocannabinoids, which travel back in a retrograde pattern and bind to presynaptic CB1 receptors, and that acts as a brake that reduces the further release of that excitatory transmitter. In the periphery, the CB2 receptor is often involved. This is a schematic of an immune cell, like a macrophage, for example, that's secreting pro-inflammatory cytokines. And cannabinoids have anti-inflammatory effects in part through this mechanism of action. So when cells release excessive amounts of these chemical messengers, the endocannabinoid system acts as a brake to help restore balance. Endocannabinoids can function at other receptors as well. And for example, the endocannabinoids and certain phytocannabinoids can bind and desensitize an ion channel on certain neurons, known as transient potential vanilloid type 1, formerly known as the vanilloid receptor type 1, and this helps reduce the perception of pain. The endocannabinoid field is moving beyond a historical focus on CB1 and CB2 and looking at a diverse complement of ion channels, nuclear receptors, other G-protein coupled receptors, and enzymes as very probable conduits for the actions of not only endocannabinoids, but some of the cannabinoids that we find in plants. If we're talking about hemp, we're dealing with a very complex natural product with hundreds of constituents, and that includes over 100 different phytocannabinoids, most of which still await pharmacological interrogation, and the most abundant and well-researched non-psychotropic of these is CBD, cannabidiol. And the hemp plant is very prolific in making lots of other secondary metabolites that interact with the ECS called terpenes. Beta-caryophyllene is one of these terpenes, and in contrast to CBD, it directly and potently binds and activates the CB2 receptor. It's a very unique, selective, and direct CB2 agonist. And research is exploring the significance of this interaction as it relates to immunology and other areas, probably contributes to the clinical efficacy of hemp oil, and not all CBD oils have guaranteed potencies of this unique compound. So now you have a general understanding of how the endocannabinoid system works. We've looked at a synaptic representation of how endocannabinoids act as modulators of excitatory neurotransmission, and we've also taken a quick look at the peripheral actions of cannabinoids and how they affect CB2 receptors and therefore reduce inflammation and potentially reduce pain through their actions at the vanilloid receptors. I hope this has been helpful, and thanks for listening.